Hello friends, welcome to another episode in my series on serotonin. In this video, we're going to discuss the mechanism of SSRIs as well as their major side effects. And I'm going to distinguish a little bit between the different SSRIs and what they are known for doing in terms of side effects. Now, to be honest though, this review doesn't include a comprehensive review of SSRIs, because I could include, for example, the difference in their half-lives and some other small things like their bioavailability, but this is really the useful stuff. So, and plus, by the way, if you'd like any more information, you can always look at the citations that I provide in the videos. Now, I have a feeling that there are some people that are going to view this video that haven't watched the rest of the series. I admonish you for doing this. I'd like you to know that the series is very easy to listen to, especially if you're, for example, going to the gym or doing cardio. You can put the playlist on and go through the whole series. Why I recommend doing that is that you need a full understanding of serotonin's biology in the body to really understand the value of SSRIs. And if you do listen to this series, I can almost guarantee that you will know more about serotonin than your local psychiatrist. But you will still need your psychiatrist to prescribe SSRIs to you, of course. But what I'm saying is you'll really get a very detailed understanding. Nonetheless, hopefully you've caught up to the other episodes before you hear this one. So let's talk first about the mechanism and the function of SSRIs. So SSRIs are thought to function. Now, first of all, we know that they function depending on serotonin. This is evidenced by tryptophan depletion studies using SSRIs. So if you deplete the diet of tryptophan, which is the dietary precursor of the synthesis of serotonin, the SSRI's effects are attenuated. However, there is still some, not so much disagreement, but there are developments in terms of the understanding of how SSRIs work. So if you recall from earlier videos, essentially this all began with tricyclic antidepressants, which had many functions in the body. One of them was to inhibit the activity of SERT, SERT being the transporter that takes serotonin out of the synaptic cleft where it's communicating between neurons. When you inhibit SERT, serotonin stays in the synaptic cleft, causing more serotonergic activity. So tricyclic antidepressants did that. Monoamine oxidase inhibitors did that also. And then, with the advent of SSRIs, it was shown that a similar antidepressant and anxiolytic phenotype developed among people that took SSRIs, which were distinguished from the former drugs as being very selective on the SERT transporter. So, because of this reason, they developed this idea originally that serotonin was the lower serotonin levels or serotonergic activity was the cause for depressive and anxious symptoms. Later, it was hypothesized, two things were hypothesized. First, there was a neurogenesis theory of depression and the reason why this came about is that researchers noticed that when given SSRIs, the antidepressant phenotype doesn't develop for 3 or 4 months, up to 6 months, between 3 and 6 months, let's say. In that time period, what really changes is the expression of neurotrophic factors in the brain. In the beginning, serotonin, and I'll discuss this in a little bit more detail, but in the beginning, serotonin levels are increased. But it is until the neurotrophic factors increase that people actually develop an antidepressant phenotype. So first of all, there was the serotonin levels, then the neurogenesis. There's also another theory called the cytokine hypothesis of depression, which I've described briefly before. In fact, let's get into that now. Before we do though, I'd like to mention regarding neurogenesis, it's not just the creation of new neurons. It's the creation of all kinds of new brain matter, including oligodendrocytes. Oligodendrocytes are the components of the brain that produce what's called myelin. I've mentioned this in an earlier video, but for a refresher, myelin is the lipid-rich substance that covers axons in the brain. Axons are those tentacles, so-called, I call them tentacles, they're not really called tentacles, that connect the RAF nuclei, which are the serotonergic nuclei of the brain that synthesize serotonin, to the forebrain from the rostral group or backwards from the caudal group of the RAF nuclei. These axons have myelin sheaths. The disease of having pathologically low amounts of myelin is called ALS. That's what Stephen Hawking had. Also, having genes that produce more myelin are associated strongly with intelligence. So oligodendrocytes produce myelin, and certain SSRIs like fluvoxamine increase the rate of development of oligodendrocytes in the brain. So they not only produce new neurons, but they do a lot of things in the brain. Now, regarding the cytokine hypothesis of, another thing I should mention that falls in line with this neurogenesis theory of depression is that rapamycin has been shown to inhibit the antidepressant effect of SSRIs, which is fascinating. As you guys know, rapamycin is the drug that mTOR is named after, mTOR being one of the largest or the largest growth pathway in the body. Rapamycin inhibits growth in the body, and rapamycin therefore likely inhibits growth factors in the brain. And the SSRI's effects on the SSRI's production of an antidepressant phenotype is inhibited by this reduction of neurotrophic factors. So the main, for me, the main reason SSRIs work is because of the neurotrophic factor. But there's also the cytokine hypothesis, and I want to tell you guys why the cytokine hypothesis exists. I've never done that fully on video before. I've just described it briefly. I've told people, if you inject people with inflammatory cytokines, they develop a depressive phenotype. But let's get into the details. Now, number one, this is why it developed. Number one, patients that were given treatments involving tumor necrosis factor alpha and interleukin-2 developed depressive symptoms. Number two, the phenotype caused by having higher interleukin-1 levels and higher lipopolysaccharides, which are, by the way, they're created by the gut microbiome. I shouldn't go into too much detail. Is similar to the depressive phenotype. So those are 2 points. Number three, inflammatory cytokines activate the hypothalamus, pituitary, adrenal, HPA axis. That is an axis that is overactivated in depressive people in general. Number four, some cytokines regulate the activity of both serotonin and norepinephrine. So because of these reasons, a cytokine hypothesis of depression was developed, and SSRIs have an effect on inflammatory cytokines, and some people think that its modulation of inflammatory cytokines may also be how it reduces depressive symptoms. An interesting point is that non-steroidal anti-inflammatory drugs, NSAIDs, may attenuate some of the beneficial effects of SSRIs in the brain. So for example, SSRIs actually increase tumor necrosis factor alpha, which is an inflammatory cytokine, and interferon gamma, which is also an inflammatory cytokine, in the brain, which results in higher P11 levels. P11 is a member of the S100 protein family, and it interacts with serotonin receptors. With the higher P11 levels, an antidepressant response in the brain exists that produces an antidepressant effect. By the way, mice that have a knockout of P11 are depressed, and mice that overexpress P11 have an antidepressant phenotype. So this shows you how sort of the inflammatory effect SSRIs may modulate the depressive phenotype by affecting the inflammatory cytokines. Now, regarding what SSRIs do in the brain acutely. So when SSRIs are first given, the first effect is that the RAF nuclei, which are the serotonergic neurons that produce serotonin in the brain, their activity is diminished. So the acute increase in extracellular serotonin in the frontal cortex of the brain is stopped. Although in the medial area, the midbrain, extracellular serotonin still rises by a factor of 2 to 3x. What this means is that because the SSRI comes in, the RAF nuclei realize there's a lot of serotonergic activity. So they stop producing as much serotonin, which limits, because the RAF nuclei is sent through the axons to the front of the brain, it limits serotonergic activity in the front of the brain, although in the midbrain it's still increased, just because of the dynamics of what is going on. But then in the second step, what happens is the RAF nuclei recover due to the desensitization of the 5-HT1A autoreceptors, which I've mentioned in many videos. These are one class of serotonin receptors that there are autoreceptors. They sense serotonin and they send a signal back to stop producing so much serotonin. And that's how actually the serotonin the activity of the RAF nuclei in the first stage were decreased. But then these things get desensitized, which is a key element in the physiological effects of SSRIs. They desensitize the 5-HT1A receptors, which estrogen does also, and which is a very important facet in their pharmacology. This in turn increases extracellular serotonin in the projection areas of the RAF nuclei. Projection areas means the areas that are receiving serotonin from the axons, those so-called tentacles that I call tentacles. Now, interestingly, the initial effect, which is this decrease in activity of the RAF nuclei, can actually be blocked with the use of beta blockers, because beta blockers also block the 5-HT1A receptor. So if you're starting an SSRI treatment, which many psychiatrists don't know, if you're starting it quickly and you really want the effect to happen quickly, and if the person doesn't have risk factors for it, you can give them a beta blocker, which will inhibit this downregulation of RAF nuclei and increase the speed of the process happening. Now let's discuss side effects briefly. First of all, there is the major side effect is sexual. So in general, all SSRIs seem to produce sexual side effects at a similar rate. However, there's a difference in the rate of sexual side effects depending on the kind of drug used. And the drugs are, by the way, these are the drugs that are very specific, used in psychiatry, that are used for depression or anxiety. One is a selective norepinephrine reuptake inhibitor. One is a selective dopamine reuptake inhibitor. One is a selective serotonin reuptake inhibitor. And then there are combinations of them. So there are some that, for example, are selective for norepinephrine and serotonin. Okay? So, norepinephrine reuptake inhibitors produce the least sexual side effects. They're 5 to 10%. Norepinephrine and dopamine reuptake inhibitors like Wellbutrin or bupropion, that's what it's called, produce 10 to 25% rates of sexual side effects. Serotonin and norepinephrine reuptake inhibitors produce 58 to 70% sexual side effects, and SSRIs produce 25% to 73% sexual side effects. Why I'm using those numbers, by the way, these are the lowest numbers in some studies and the highest numbers in other studies. So basically SNRIs and SSRIs produce a almost similar level of side effects. Some people think that SNRIs produce a little bit less, which means that when they inhibit the reuptake of norepinephrine as well as serotonin. But basically you can see from the change in frequency that once you involve serotonin, the side effects really go up in terms of interest in sex, okay? Also, some other things that your psychiatrist will probably never tell you. SSRIs are strongly associated with a loss of bone mineral density, of up to 70% increased risk of fractures. This is something you must keep in mind. Something probably not very relevant to those people that weight train, but something that's not talked about enough. Also, many SSRIs are associated with weight gain, and this very much depends on the person and their genetics. But I want you guys to note some things. Paroxetine is the worst for weight gain among the classic SSRIs. I mentioned mirtazapine before, but that's a selective norepinephrine and serotonin reuptake inhibitor, so it's not included here. Also, many SSRIs cause gastrointestinal distress. The worst for this is fluvoxamine, which is the SSRI I like. The best, probably with the least side effects for this, is escitalopram. In terms of changing the QT interval of the heart, usually make it slower. Anyway, in terms of changing the QT interval of the heart, citalopram is the worst, and paroxetine is the best. When I say best, means it has the least effect on this. In terms of liver injury, for those that have very compromised livers, citalopram and escitalopram appear to be the safest. Now, I don't know whether I should continue this in this video or not. Yeah, let's do it in this video. Let's discuss the genetics of serotonin reuptake inhibitors' effectiveness as well as their metabolism. So a lot of people talk to me and say that, I get really frustrated when people make comments on the videos like, and I see this on Reddit a lot also, it's not even on my, on the nootropics section. People demonize SSRIs. They demonize them. Some people say that SSRIs made them want to kill themselves, and of course, all these people always took SSRIs for like 2 weeks while they were depressed, of course, So who knows what's going on with these people? But anyway, the point is the effects of SSRIs or the effectiveness of SSRIs is mediated by genetic factors, okay, and the individual pharmacology of SSRIs is mediated by genetic factors as well, specifically polymorphisms at the P450 cytochrome enzymes. So I'm going to discuss these briefly with you so that if you want to take SSRIs, you can look into the different structures of the different SSRIs and your genetics, if you don't want to have yours analyzed. In fact, to be honest, I don't look at these too much myself, but here's what you can know. There are two particular cytochrome P450 enzymes, which are called CYP450. There are two that are particularly relevant. One is called CYP2D6. CYP2D6, which I'll continue to call the 2D6 from now on, has over 100 allelic variants and subvariants, which are... Sorry about that, folks, I had a technical difficulty. Anyway, the 2D6 has over 100 allelic variants and subvariants that are relevant and found in quite high frequency in people. There are three major groups of them. One that causes decreased enzymatic function of the CYP2D6 enzyme, one that causes normal function, and one that causes no function. I'm going to list the genetic terminology for these groups, and you can research them on your own to find out which one you may belong to. So, for normal function, the CYP2D6, I don't know how to describe it actually. It's like a star. You can find it if you do Shift and 8, I think, on your keyboard. But this top star thing, that's how they're described, with the star and then a number. So the star 1 and star 2 produce normal function. The star 9, star 10, and star 41, which you can Google, just search CYP2D6, then that small star at the top, then a number, and you can Google them. The star 9, 10, and 41 produce decreased function. The star 3, star 6 both produce no function. Star 5 is actually a gene deletion. There's another enzyme of the CYP450 enzymes that is very relevant. It's called CYP219. This one has over 30 allelic variants and subvariants. Most of them belong to three groups. Most people belong to three groups. One produces normal enzymatic function, that is CYP219 star 1. Star 17 produces increased enzymatic function. Star 2 produces no function. You probably belong to one of these three. Now, let me tell you how this is relevant to the pharmacokinetics, which means the metabolism of and drug action over time of the SSRIs. So I'll just briefly summarize. Paroxetine and fluvoxamine are both metabolized by the CYP2D6 enzyme into compounds that are inactive for the result on SERT. So if you have a higher activity of CYP2D6, you will need quite a bit of a higher dose of fluvoxamine to get an effect, because a lot of it will be metabolized into these inactive compounds. So if you have a very high activity of CYP2D6, you may not want to use fluvoxamine or paroxetine. Now, fluoxetine is metabolized by both of the enzymes, CYP2D6 and CYP219, into norfluoxetine, which is pharmacologically active at SERT. So it doesn't really have an effect on the activity of SERT. Although you can compare norfluoxetine to fluoxetine to see the differential effects. I'm not quite sure of the details of them, but it is active. Citalopram and escitalopram are both metabolized by CYP-C19 into a less pharmacologically active compound. So if you have an overactive increased function, so if you're, for example, the star 17 of CYP219, you're going to have less of an effect from lower doses of citalopram and escitalopram. In terms of sertraline, it's metabolized by both of the enzymes, but CYP219 appears more relevant to its metabolism. Now, I should mention briefly, because I'm not going to mention it later, not all of these SSRIs have and I didn't write this actually in my notes, but not all of these SSRIs have the same pharmacologic profile. So they all inhibit SERT, but some of them do other things as well. One of the things that they do is agonize the sigma-1 receptor, which is also agonized by ibogaine and several other drugs, including potentially, for example, kratom, I think, and many drugs. Many of the psychoactive drugs agonize the sigma-1 receptor also. And we don't really fully understand the sigma-1 receptor. But fluvoxamine appears to agonize it the most, and that's the drug I take. And I do believe that the sigma-1 receptor has some effect that is beneficial. There is also another issue when you're considering an SSRI, which is the health effects of them. So I described in the cardiovascular and cancer section some of the, maybe briefly I described them, but some of the drugs are more associated with health outcomes than others. For example, I don't think that fluoxetine, which is Prozac, has a very favorable research outcome, but the issue is that fluoxetine is more researched than the other ones. In terms of side effects, we know, I just described them to you, but in terms of cardiovascular effects or effects on fibrosis or things like that, a lot of it depends on the metabolism of these drugs and their other effects in the body. So it's a little complicated. But anyway, this series was not meant to be a guide on how to pick an SSRI. I'm adding this just sort of as notes. It was meant to be a guide on how serotonin affects the body and how to think of serotonin. But I'm adding this anyway. Finally, I want to talk about the effectiveness of SSRIs as they depend on your genetic polymorphisms. And this is going to be a little detailed. I'm not adding this here for my own sake. I'm adding this here because there are some people that watch my videos that like to do their own research into their genes, but cannot afford to, for example, pay me to do it for them. And sometimes they ask me to do videos including like individual SMP numbers so that they can research them, which the only reason I don't do those videos is because it would, first of all, I'm not interested in doing those videos. It's a little intellectually boring for me. It's basically the results of genome-wide association studies. And second of all, they will get very few views. So they're only going to help a few people. And those people, basically, it's helping them because they can't afford to pay for the research that the products that I do, which I don't mind doing, but I can't spend the time to do that. But while we're on the topic here, I'm going to include some of the, basically, I'm going to include my notes on genetic polymorphisms that affect SSRI results. But these are not my notes on the serotonin system in general, because that's way too long to go over right now in terms of SNPs. But I'll just describe the ones that seem to have an effect on SSRI effectiveness. So to begin, I just want to list the genes for you. Other than the P450 genes, which are the enzymes that metabolize the SSRIs, there is the SLC6A4 gene. And in addition to that, that's the SERT gene, by the way. In addition to that, there are other genes related to that, like in the promoter region and so on. There's the HTR2A gene, which is the serotonin receptor 2A. There's the BDNF gene, which is the brain-derived neurotrophic factor gene. There's the GNB3 gene. There's the FKBP5 gene. There's the ABCB1 gene. Now, to begin, the SLC6A4 gene codes for SERT. That gene, as well as 5-HTT-LPR, which is the promoter region of the SERT gene, and RS2553 and ST, capital letters ST, lowercase in2, are the most researched in regards to the effectiveness of SSRIs. So, people who have a short allele at the promoter region, which I've described in an earlier video, or a long allele with a G at RS25531, to RS25531, respond worse in Caucasians to SSRIs. In Asians, S's, which is the short allele, may respond better. In terms of the serotonin receptor 2A, HTR2A, there are 3 SNPs that are well studied, but the results are unclear. If you want to look into them, they are RS7997012, RS6311, which is a very famous SNP, and RS6313. At BDNF, RS6265 is the most studied, and heterozygous genotypes as well as the MET carriers respond best to SSRIs. The GNB3 gene codes for the G protein polypeptide 3, which is a second messenger system involved in a lot of activities. RS5443 is the most studied polymorphism in relation to SSRI results at the GNB3. Better results are found in carriers of the T allele in Caucasians, but maybe not in Asians. And this again speaks to the polygenic complex effects of, you may have, the effects of one allele depend on your genetics in general. I've discussed this in an earlier video, but that's why ethnicities respond a bit differently. So, The FKBP5 gene, which is responsible for AKT activity and glucocorticoid receptor sensitivity. There are several polymorphisms that have been associated with SSRI effectiveness. One is RS1360780. The TT, that means homozygous T genotype, has been shown to have a faster response to SSRIs as well as a quicker HPA response to the SSRI. So it's the hypothalamus pituitary adrenal reconfiguration in response to it. Also, the RS3800373, the C allele there, the RS47139116, the A allele there, and RS352428, the G allele there. These are all associated with better results among from SSRIs. Finally, ABCB1, which is very much involved in blood-brain barrier permeability and affects the if it has, like, for example, it doesn't affect some drugs, but it has it's responsible for the ATP dependent drug efflux pump from some other drugs. So for example, it doesn't affect fluoxetine and for example bupropion, which is Wellbutrin. Anyway, this gene, there are 3 SNPs there which seem to affect many SSRIs effectiveness. One is RS2032582. The TT genotype seems more effective here. RS2032583, the C allele here, and RS2235040, the A allele there. These all seem to increase SSRI effectiveness. So I think I'll leave this here for now, and then in the concluding video I'll talk a little bit about what this all means. Alright guys, thank you so much, and I'll see you next time.